Ladies and gentlemen, it's a great honor to receive this award, and an even greater honor to stand here today, because it was actually on this very conference 7 years ago when I decided to step onto a scientific path. I hope many of you will be inspired similarly after this conference. My research lies in the intersection of mycology, evolutionary biology, and developmental genetics. So this field is fungal evolutionary developmental biology, or in short, fungal evo-devo. I know this is mainly a biomedical conference, and I believe most of you are interested in medical sciences. Still, I'm here to talk about basic research and fungal evo-devo, so some of you might be skeptical. Some of you may feel a lack of faith in it. Well, I will do my best to convince you that fungal evo-devo is also very interesting. Let's start with the evolution of complexity. 3 billion years ago, life was quite simple, and I always found it astonishing that from these very simple cells, evolution was able to build these giants you can see here, including mushroom-forming fungi that include some of the largest organisms on Earth. Contrary to popular opinion, this increase in complexity is not the only tendency in evolution. We can find several examples of the opposite trend. Here you can see a reduced crab species and the yeast species, which is actually a simplified fungus. But my research focuses on complexification. The highest level of complexity, what is called complex multicellularity, can be found among animals, green plants, red algae, brown algae, and fungi. These organisms exhibit a three-dimensional architecture, a diversity of cell types, and a developmental program. What does it mean? A developmental program literally means what you can see on this video, that each and every cell knows exactly what they have to do, when they have to divide, when they have to differentiate, in order to build a properly functional structure. These programs are encoded in the genome and thus passed from one generation to the next so they can evolve. The evolution of these developmental programs is the focus of the field of evo-devo. Mushroom-forming fungi represent simple multicellularity and complex multicellularity at the same time. So here you can observe the striking difference in organization between the simple mycelium and the complex mushroom. The evolution of complexity was mainly studied in animals and plants. All the other groups are understudied. And one widely accepted principle states that achieving this high level of complexity is so difficult that it can happen only once or just a few times in a lineage. This is true for these 4 taxa, but fungi break this rule. They evolved complex multicellularity at least 8 times independently, and they defy other principles as well. So without fungi, we won't be able to fully understand the evolution of complexity. I've brought you some practical considerations as well. The mushroom industry is constantly growing, and fungi have tremendous potential that is currently underexploited because of the lack of sufficient knowledge. For example, there's promising research on fungal leather substitutes and even mycelial bricks. And if we want to enhance their useful traits, we have to get to know their developmental genetics better, because throughout the millennia, humans were able to radically transform plant species and animal species as well, but for some reason it didn't work with fungi. So for further progress, we need solid genetic knowledge. Now I'd like to show you some aspects of my own research. In the bottom right corner, you can see our model species, an inky cap mushroom. This species can be genetically modified and has a very short life cycle. In my master's studies, I made research on 2 RNA-binding protein genes. RNA-binding proteins are known to be critical in animal development, and we were wondering whether a similar layer of regulation might exist in mushroom formation. By comparing expression data in a simple species and a complex species, we selected these two genes because their expression was intriguing from an evolutionary perspective. Upon overexpression, we observed branching fruiting bodies as rare but reproducible phenotypes. So I named the genes after the multi-headed dogs of classical mythology. One of them is called Ot2 after Orthrus, the two-headed dog. The other is Cer3 after Cerberus, the three-headed dog. You can remember these figures from the stories of Hercules. And in our lab, we also have Snowball and Gandalf phenotypes. So, dear students, if there's just one thing you will remember from this presentation, let it be this: make sure you never choose boring gene names in the future. So Ot2 and Cer3 are actually the first RNA-binding proteins with a demonstrated function in fungal complex development. And we summarized our results on Ot2 in a publication. Our model species forms a different kind of structure in darkness. Let's call it dark-grown fruiting body for now. And we showed that Ot2 mainly plays a role in the formation of this structure. Without the Ot2 gene, the fungus didn't form any dark-grown fruiting bodies. However, the overexpression of the gene resulted in significantly more of them. This finding can be useful in mushroom cultivation, since there are some species like enoki, which are specifically consumed in their dark-grown forms. In my main doctoral project, we would like to investigate mushroom development using single-cell RNA sequencing. Let's see what it means. In the case of bulk RNA sequencing, so when we just take a tissue sample, extract the RNA, and sequence it, we see only the average gene expression. But sometimes the information we seek lies in the differences between the individual cells. So if we are able to sequence the RNA content of the individual cells separately, we see something like this. The beautiful colors do not average out. This is a famous painting. I'm wondering whether you know which art movement it belongs to. If we zoom in, you can see the points, the individual cells themselves. So this is a pointillist painting, actually. And this is another pointillist painting, the cell atlas of Betwing from a paper I've recently read. By comparing the transcriptomic similarities of the individual cells, we can find different cell types and we can assess their relations. In the past, when researchers annotated cell types exclusively based on morphology, they usually underestimated their numbers. But now we know that humans, for example, have more than 400 cell types, and the most widely used plant model has almost 200 cell types, but we don't have any transcriptomic data on how many cell types mushrooms might possess. The reason we are so interested in cell types is that they represent innovations in the evolution of complexity. We would like to create similar cell atlases of 4 fungal species representing 3 different levels of complexity. The species on the left is a quite simple one. Its sexual structures are microscopic. The species in the middle has a fruiting body of 3 cell layers, and you already know how complex mushrooms are with different tissues and cell types. By comparing these cell atlases, our aim is to uncover the evolutionary origin of these different cell types, these innovations of the evolution of complexity. I'm really grateful to my mentors, László Nagy and Árpád Csernetic, for their guidance. Árpi, thank you for being here. And I'd also like to thank my secondary school teacher, József Gőz, for setting me on this path 7 years ago. Thank you for your kind attention.
Thank you very much, Benedek, for this excellent presentation. And I would like to ask the mentors as well to come up. László Nagy unfortunately cannot be here, but the junior mentor, Árpád Csernetic, is here as far as I understand. Yes, he is coming. Please come over, and we would like to provide the award for you and for the mentor as well.